We have school these days. No. Continue. O-level. Long. Question number 8. Guanine, adenine, cytosine, and thymine. So base pairing is guanine with cytosine, adenine with thymine. Explain the role of these bases in the structure of DNA. Use the information in the diagram to support your answer. I think it's very clear. DNA molecule, 2 strands joined together by making hydrogen bond, guanine with cytosine, adenine with thymine. So they make the 2 strands bring together by making hydrogen bond, and base pairing is guanine with cytosine, adenine with thymine. If you want, you can still write A and T make 2 hydrogen bond, G and C make 3 hydrogen bonds. This is called complementary base pairing. Complementary base pairing. The DNA of bacterium E. coli has mass of 3.1, 10 to the power 9, Dalton. The mass is this much. Base pairs 4.7, 10 to the power 6. Calculate the mean mass of base pair. Express your answer in standard form. So mean mass mean there are 2 base pairs, A with T, C with G. So we get the mean of this. So we have to divide this mass with the base pairs. So 3.1 divided by 4.7 multiplied by 10 to the power 9, 3. So what is the number you get? 659.5. So you have to express in the standard form 6.5 but you have to round up 6.66, right? 6.66. Six 10 to the power 2. Mass of base pair. Calculate the total mass of DNA produced after 3 cell divisions. Okay, 3 cell divisions, 1 cell is a 1 cell division. And then 2 cell division, and here is a 3 cell division. So basically, it should be 2 to the power 3. 8, right? 8 cells. After 3 cell divisions, we have 8 cells. And 1 cell has 3.1 multiplied by 10 to the power 9 Dalton. This should be multiplied by 8. $24.1, no, 24.8, 10 to the power 9 is a total mass. So 1 Dalton is 1.6 10 to the minus 25 gram. So 1 is this much, and how much is 1 Dalton equal this? So we have a 24.8 10 to the power 9 Dalton. This should be multiplied by 1.67 10 to the power 24 gram. If 1 Dalton is this much, 100 Dalton is this, like that, right? Ooh. So minus 24 plus 9 is using the let's say like this, 10 to the power minus how much? 5, no? 15, right? 15, 10 to the power minus 15, 24, 8, 1.67. So we had to further multiply this number. Calculator. 248, 24.8, 24.8 multiplied by 1.67. So I write it here, 41.416 minus 15 gram. So we can write in a standard form then. So 4.116 gram. Hmm? 16, no? Because this decimal is moving here. This should go this direction, right? Backward mean 15, 14. Yeah, it's 14. So this way is getting 16. No? Yeah. Now my question is, can you do it in a calculator, this one? Is it possible to do this with a calculator? Hmm? Yes. Okay, if you can do it in a calculator, you can do it, right? Because you get a more accurate result. Otherwise, we have to go to the rules like this decimal, for example, this decimal point is moving forward. What we remember, the power is going this direction, opposite direction. Opposite direction is in a negative scale, it is minus 14 because negative scale minus 15, minus 14, minus 13, minus 12. Going this direction mean it is getting smaller and this direction is getting higher. So we can make easily mistake that I made, right? So if you can use the calculator for minus 10 to the power minus 24, for this one, it's okay. And you get this kind of a very big numbers for calculation sometimes. You have to get used to this kind of a calculation. You get huge numbers to calculate. These are the two guys. I think we talk about Michelson-Stahl experiment. So one of the picture of these two guys about this semi-conservative theory, not theory, semi-conservative experiment. Stage 1, stage 2, and stage 3. I think you should be very familiar now. These numbers. First generation, I mean, first N15 medium, everything is heavy. Then change to the N14 medium, then it's get a hybrid version, N14 and N15. Then it's going to the next medium, it get the light and hybrid version. I hope you can remember this, how it appears in the tubes, right? So this should be very well remember, otherwise it's here, and the next one is in the middle, and this one, it's in the middle and here. 50-50. 100% hybrids, 50 in 15, 50 in 14. Hybrid, right? Hybrid. And 14 is 50%. After stage, okay. Complete the diagram to show the result of this investigation. Use the dotted line to help your position to band. I think initially, when we doing this one, we try to remember it as a maths calculation. But at one point, we try to remember the band's position, everything, numbers, because we have done many times. We can remember it. It's easy for the exam if you can remember it. Stage 1, it is growing only with N15 medium. It is only heavy bands. Then change to N14 medium, first generation. This is the first one they according to this first, and this is the second one. And second one, what we observed somewhere here, hybrid version. Because N14 and N15. Next one, here one band and here one band. Better remember 50% light and 14. This one is 50% light and heavy. That is N4, N15 and N14. This one is a hybrid N14, N15, 100%. So it is better that you make a small, you take your notes and try to remember first, second, third, and fourth. What is the band pattern and percentage? Now this is the one Messelson and Stahl did to prove that the DNA replication happened in semi-conservative method. But before that, scientists also proposed other models. What are the other three models? Conservative, dispersive method. Conservative method, scientists proposed the old strand remain as like this. Here, this is the old strand, and old strand make a copy, new copy is formed, and old strand stay like this. And from this one, old strand stay here, and old make again a new one, and this one is also like this. So old strand remain as a template, and then rest of the stand, like from this case, if you take this one conservative method, uh, there is no semi-conservative, so this remain as a old, this is a new strand, this is a old, this is a new strand, so it is like this. And dispersive method, in both the strands we can see mixture of N14 and N15 like this. It's always get same thing. So if you get always same thing, we should see same one band. This is a conservative theory, old, new. DNA after stage 1, and then after stage 2, we can see it. This is stay here, and this stay here. Old and new. There's no intermediate forms. Right? And the dispersive method, everything after second generation same. That means you can see here there's no old one, so then everything is somewhere here same. The band is like this. It's a mixture of both heavy and light all the time. Okay, now I just wanted to know, if you add these 5 marks, we have done 4 questions, right? Now let me check how many marks you can get if you have done these 4 questions. Before that, we want to check what is the total marks for this. Can you calculate total marks for these 4 questions? Fifty? Huh? Yes, yes, 5 to 8. 5 to 8, yes. So you said 50, right? Okay, question number 5. Now, question number 5, question number 6, question number 7, question number 8. So if you add these 5 questions, you get a 50 total. Right? 1, 2, 3, 4. Now I suggest I very the general for all the students, right? I'm suggesting for all the student, any student, they should maintain at least 30 for this. Yes. Question number 5, 6, 7, 8. Now, out of 50, all the student can maintain 30, close to 30. That would be great, because now you have how many 30 left? You have 30 left for the 1, 2, 3, 4 questions. So 1, 2, 3, 4 questions, their difficulty is not much. They're not that difficult. That means you should at least get 20. So then if you have 50 out of 80, so far the UMS and the Romax conversion, uh, you are in the A. You can get A. So how many marks you get out of 50? 30? Oh, good. So now thing is, if you join online and we don't have much communication, it's very hard for me to make some kind of a guarantee or some feedback to you how you ended up in your exams. But when he's here now, he get 37 out of 50. So this is a good mark to get an A, because if we consider any paper is a standard paper, and if you get the same paper in the exam, if you can maintain more than 30 for the last 5 questions, you can get an A. But I cannot evaluate you because you are in the other end, and I don't know what you because you have done. But the paper, send it to me. I think Gitmini, you can self-evaluate, right? You send me the paper, so you can also check how many marks roughly you get. I mean, as we marking, it is not exactly 37. It could be here and there. It can fluctuate certainly. Let's say we give plus or minus 4 or maximum 5, plus or minus 5. There could be variation like this. So still, if we are in a range, we are close to 30, it is safe. So then we can continue for the last 4 questions, targeting to get more than 20. Otherwise, variation of the exams, because sometimes paper is bit tricky, new questions come, you can get marks, you can lose mark. But we are safe if you are in this range, above 30 and above 20. So we are exceeding above 50 then. Yeah, you got 30, right? Tisara, you got 30. Yes, if you have 30, it's okay. I mean, you build up your self-confidence. I mean, that's also one thing that you have to stronger, no? Otherwise, you always panic. You are always worrying about the exam. You might thinking, should I get A? Should I get lower mark like this? But it is something that when you get the marks, this is some kind of a guarantee for you that you are in a safe region, and you should not stop working. You have to work, but you have a little bit of less stress now. At least I am in a safe region. Still, I am going with your marks. So that's very important. But I don't know the now I mean, self-evaluation is okay like this. But other Jitmini, I don't know whether you have evaluated your paper. Anyway, you joined a little bit later. I know that. Uh, so we go for question number 2 then. We check quickly some of the questions in question number 2. What is the name of blood vessel Q? So blood vessel Q, I think you can easily guess it, because the arrow is going to the right atrium. That could be vena cava, right? This should be vena cava. And that's a very easy question. Cardiac cycle has atrial systole, ventricular systole, diastole. Now you see, they are not that difficult, right? Directly asking question. The duration of ventricular systole is the time taken it takes for the impulse to pass between E and P, shown in the diagram. Calculate the proportion of time that the ventricles are contracting in one heartbeat if this heartbeat 73 times per minute. Now here E to F. The duration of ventricular systole is E to F. That means ventricular systole is time E to F. That is 0.0 is 0.05. Right? 0.05. So E to F, duration of ventricular systole is time taken for the impulse to pass between E and F. So E and F 0.05. Calculate the proportion of time that the ventricles are contracting in one heartbeat if this heartbeat 73 times per minute. If this beats 73 beats per minute. So this should be second then, right? It's not mentioned. The duration of ventricular systole, time taken, impulse to pass between time taken. So time taken should be given as second then. It should be. Yeah. 0.05 second taken for one ventricular systole. One ventricular systole 0.05 second. So one ventricular systole time taken 0.05 second. Then how many for 73 ventricular systole? Because heartbeats means 73 times ventricular systole. 3 point. That can't be. Because here 60 second. 1 minute is 60 second. Proportion of time that the ventricles are contracting in one heartbeat. So total we get 3.65. Second, 7, okay, 73.6 beats takes 60 seconds, 60 seconds, 60 seconds. So one beat, 60 divided by 73. One beat is 60 divided by 73. 60 divided by 73, 0. Roughly 82 seconds. One cardiac cycle is 0.82 second. One cardiac cycle is 0.8 second. We learn one cardiac cycle is 0.8 second and 0.4 second is diastole. 0.1 second is atrial systole. 0.3 second is ventricular systole. So these are the standard forms. One cardiac cycle in average heart, like a resting heart, 0.8 second. Diastole this, systole, ventricular systole this much, and atrial systole is this much. So here, according to this question, 0.05. The duration of ventricular systole is time it takes for the impulse to pass between E and phi, E and F. So E is here, F is here. Calculate the proportion of the time that the ventricles are contracting in one heartbeat if the heartbeat 73 times per minute. Okay, let's do like this then. 73 cardiac cycle of heartbeat in 60 seconds, right? So let's say heartbeat. So time taken for one heartbeat is 60 divided by 73. So this should be 0.8, let's say roughly, if you divide 60, 73, 0.82, right? Now one heartbeat, it takes 0.82 second. According to this, duration of ventricular systole is the time it takes for the impulse to pass E and F. E and F, the time is given 0.05. Proportion of time that the ventricles are contracting. So this is total for all heartbeat, and ventricular is 0.05. This is 0.82. So should it be then 0.05 divided by 0.82? Hmm? If you make 100, you get a percentage, right? This is time for one heartbeat, time for ventricular contraction. So if you want to get the proportion, 0.05 divided by 0.82 into 100. It gives the answer 6%. So you have to calculate the speed between B and D. B and D, the distance is 12.5 millimeter. Speed is 0.12. So speed, distance divided by the time. So this is, what's a 100 millimeter per second, right? 104 millimeter per second. Distance, speed equal distance divided by time. Speed need to be slower than the speed that the impulse travel between other part of the heart. Okay, why this speed should be slower? First, atrium contract. After atrial contract, then the signal is going to the ventricles. So it need time completely empty the atrium to the ventricles. Then only ventricle contract. Right? So answer is it need time to contract the ventricles. It need time to empty the blood into the ventricles. So atrium contract and blood empty into the ventricles. So there is a time for that. Then the ventricles start to contract. So here it is slow down and then it goes back. Then the ventricle can contract. So the contraction signal is going first to the atrium contract, slow down here. Once atrium contract over, then the ventricle contract. So yes, I think we have only done only done 8, 7, 6, and 5, 2, 1, 3, and 4. We couldn't done. Okay, I can stop. We'll see how we can manage for the next few days until the exam. So I'm going to stop and let's see what we can do for the next round, right?